Hello everyone, a very warm welcome to my presentation. So let me take you all back to 120 years in Frankfurt, Germany, where this physician by the name of Alois Alzheimer was practicing when he encountered this patient who was suffering from memory problems. The more he worked on her case, the more he realized that her condition was extremely unique, and even after her death he kept on studying her brain tissue samples and found toxic protein aggregates called plaques and tangles, which even to this day are the major diagnostic biomarkers for Alzheimer's disease. Unfortunately, in India, we do not really have AD records. However, we do have dementia records. And based on that, you can imagine millions and millions of Indians suffer from AD. And in rural India, the condition is obviously much worse, where the level of education is also much worse. Now what happens is in the healthy brain, due to these proteotoxic aggregates which Dr. Alzheimer found, the cognitive decline ensues due to the neurodegeneration. And even after years of research, we do not really have any proper cure. So death is almost evident. Moreover, we do not really understand why women are more likely to develop this disorder. Now, if you go back to 12th standard biology, females have this extra X chromosome, right, which males do not have. And evolution does not really like anything to be dissimilar. So what happens is females also have this extra RNA called XIST, which males do not have, which inactivates this extra X chromosome, which can be seen under a microscope as this bright spot known as the Barr body. Now, if you see in case of female AD patients compared to the healthy controls, this RNA XIST is also significantly upregulated in female AD patients. So this begs the question, because since men do not really have this RNA molecule, whether studying this RNA could hold the key or could help us understand why female are more prone to this disorder. And that is precisely what we have tried to do. In an environment full of proteins and RNAs, we have tried to find out the role of one particular RNA, almost like finding one guy in a platform of thousands and thousands of moving people, only worse, right? So in the nucleus, via a complicated chemical reaction, that extra X chromosome is inactivated. However, this mechanism is severely damaged in AD. So here XIST sort of acts as a savior. It gets into uncharted territory, that is the cytoplasm, almost acts as a protective barrier so that the predator microRNA cannot degrade the prey mRNA and it can't be eaten. So the supply of the proteins that are required inside the nucleus can be maintained and this light can be kept on. But how XIST itself gets to the cytoplasm? Want to find out? Check out this QR code. Thank you.